Thanks very much, Ned. It's a pleasure for me personally to return to this wonderful production facility you have here, and for SNID. It's my honor as immediate past president to thank you also for the opportunity to work in collaboration with the people here. Now, I would like to take this opportunity to mention that this two-day course is perfectly in line with the mission of SNID, which is to foster the appropriate application of these technologies in drug development. For these 4 2.5-hour sessions, we will be addressing specifically positron emission tomography, but this actually has gone so well, and we're so grateful to the subscribers that we would like to do the same thing for MRS and MRI, perhaps in another 9 months or a year's time, and then perhaps other imaging technologies as well. So thank you very much for this opportunity to put on this show today. Now, what you'll be seeing this morning will be an explication of the physics and the chemistry and the kinetic modeling, which is inherently powerful in the use of positron emission tomography. In fact, PET allows some of these treatments of the data in a unique way because of the physics of positron emission tomography. And I would like to point out in particular, what I've done is I've plagiarized a couple slides from the speakers in each one of the sessions in order to make some key points. And what I'd like to make clear, first of all, is that the coincident radiation you see indicated by arrows going in 180-degree opposite direction from the annihilation of the positron, this coincident radiation is something which is unique to PET and gives it some of its power. Now, the speakers for today will be Bill Moses from Berkeley, Henry Van Brocklin, also from Berkeley, and finally, Rich Carson from the NIH. I'd like to introduce also, I mentioned that I'm the immediate past president of SNID. The current president is Bill Eckelman, runs the PET facility and the clinical center at NIH. Tim McCarthy at Washington University is the secretary, and Terrence Hamill, our treasurer at Merck. And so you can see by these people's affiliations that SNID represents a coalition or a collaboration among the 3 legs of the stool: industry, academia, and government. I will be making a few comments now to put the science in perspective of drug development, which after all is the main goal of SNID. And before I do, I should mention that all of my comments are my own and do not represent any commitment on behalf of the FDA. I will be referring to FDAMA and some other regulatory issues. Another thing I'd like to emphasize, and this slide I've plagiarized from Henry Van Brocklin, is the nature of the isotopes that we use means that these truly are isotopic substitutions and not heterotopic substitutions. And therefore, whether it's a drug to be administered exogenously or whether we want to label an endogenous compound, it's clear that the compound differs minimally from the compound that we want to use as a tracer, because we substitute C11 for C12 and oxygen and nitrogen. There are other labels as well, such as fluorine, but I do want to emphasize the isotopic nature of these substitutions. And finally, I would like to emphasize in this slide, which I've plagiarized from Dr. Carson, is that the fundamental assumptions in analyzing these data, remember, we're looking at graphs of data, we're looking at things in familiar units of measure. We're not looking at funny fuzzy nuclear medicine slides. The manner in which these nuclear medicine data are converted into graphs and numbers is perfectly analogous and in fact in some ways perhaps simpler than classical pharmacokinetic modeling. Now, to proceed to putting this in perspective of drug development, I would like to emphasize that this really is a relatively new opportunity, and we expect this technology really to mushroom in its utility. And there are a number of factors contributing to this, not least of which is the development of many more tracers in the last decade. And so the power and utility of this tool in your drug development programs will be that much greater because of the basic science which has made so much progress in the last few years. During these sessions, you will see also a discussion of animal scanners and whole-body human scanners, which gives us the flexibility to scan the full range of species. And then finally, it's not unimportant that we have some flexibility within the regulatory bodies which not only makes it possible to run these clinical studies, but also to make use of these data in our dossier. I would like to emphasize that among the various imaging technologies, historically we've had good technology for anatomic investigations, and in fact ultrasound has come a long way as well, and very small structures are visible. But rather than just a quantitative improvement, what we're looking at with positron emission tomography is a qualitative enhancement of our power to look at molecular and cognitive issues as well. There are some disadvantages to PET, the radiation exposure being the most obvious one. There is some time required to develop new tracers if this is what's necessary for the drug development program. The validation is something I will dwell on to some extent in the slides just to come, but this is not anything unique to positron emission tomography. It's something common to all biomarkers or surrogate endpoints. And then finally, there is a certain amount of infrastructure required, but in many countries of the world, this infrastructure is in place already. We've talked about the disadvantages. I'd like to emphasize actually the reasons for using PET include advantages, such as not only is this quantifiable, but the results can be expressed in units which are familiar already from classical pharmacokinetics and pharmacology. The attenuation correction can be exact using both internal, but as regards a clinical study, also external standards. The resolution is quite good. Once again, isotopic substitutions rather than heterotopic. The possibility to do repeat measures. This is a non-invasive technique and therefore minimally perturbing the system. But also one of the real strengths is the short half-life of positron emitters allows us to make multiple investigations, for example, during a single 24-hour period to look, for example, at the classical plasma profile of the drug. I've mentioned the minimal perturbation already. And then finally, and this is truly powerful when it comes to issues like biomarkers and possibly surrogate endpoints, and that is the mechanistic relevance. We're not simply looking at structural changes, but instead actually looking at metabolic activity or receptor binding, etc. This is just a bit of a cartoon to emphasize the point that historically, going back 25 years or so, we tended to correlate drug effect with the dose administered. The pharmacokineticists over the past couple decades have done a very good job of convincing us that there's much greater value in correlating drug effect with exposure, that is, drug concentrations in the central compartment, whether for safety or for efficacy purposes. What positron emission tomography allows us to do is actually correlate these drug effects with binding at the site of action. And the obvious benefit to that in the human species is quite great. Now this slide, perhaps the font is too small to be very easily viewed on your screen, so I would refer you to the hard copy. These spell out why, for business purposes, positron emission tomography is quite useful. I would mention in particular the portfolio management and the project management. We may learn something with a lead compound, which then can be fed back into the backup or the follow-up project. And therefore, it's not only for the lead compound that knowing the receptor binding or the metabolic effect would be useful, but also to contribute to the ongoing non-clinical development of backup and follow-up compounds. It also provides an objective way to look across the portfolio so that the choice of development projects in this environment of limited resources can help us to manage the opportunity costs of perhaps having chosen the wrong compound to go forward. The regulatory benefits, I will talk a little bit about surrogate endpoints, but in fact the hurdle for achieving a surrogate endpoint status is quite high, and rightfully so. And so I would like to dwell more on the opportunity within FDAMA to use these endpoints as confirmatory evidence and therefore perhaps allow a single phase 3 clinical trial rather than 2 for approval. So in summary then, what PET allows us is better decisions sooner and cheaper. I would like to portray the use of these endpoints or biomarkers as having value across the drug development process, so that in the non-clinical phases we might characterize this as a biomarker. When we come into phase 1 clinical trials, it might confirm the mechanism of action or even establish a dose range. In phase 3, it may satisfy the criteria for confirmatory evidence and allow a single phase 3 study for approval, and then finally the highest level, a surrogate endpoint. This is just a cartoon to emphasize that both for pharmacology becoming efficacy and for toxicology becoming safety endpoints, these markers may serve either goal. Once again, the font is too small on this, but you have this in hard copy, and in fact I have these from Greg Downing, who's established the working group within NIH and FDA in order to achieve this common understanding of what these terminologies mean. I would like to emphasize that because this is actually gamma radiation that we're measuring and not the positron itself, it's possible to use some standard techniques already available in vitro, and using the same tracer, it's possible to go right from in vitro studies in Petri dishes down to phase 3 studies using the identical tracer. And furthermore, I would like to emphasize that even the choice of a lead compound from the chemical series might be made based on clinical data rather than non-clinical data if you can bring more than one tracer into the human in the early phase 1 studies. When I write down what I think are the criteria for the ideal clinical pharmacology tool, I find that positron emission tomography satisfies each and every one of those. In particular, I would emphasize that the statistical power of being able to do within subject studies is something which makes what might otherwise be an expensive technology really quite cost effective, because the numbers of subjects required to do these studies and to get a useful answer is much smaller by virtue of the within subject studies that are possible, once again due in part to the short half-life of the compound. Now, this is why I put the FDA disclaimer slide at the very beginning, because I want to put my own understanding to FDAMA. In particular, we know now that legislation has been passed which allows FDA to consider a single, adequate, and well-controlled study. Why is that? It's because progress, in particular in clinical pharmacology, has led to much better understanding and therefore fewer erroneous conclusions in the drug development. And therefore, this single, adequate, and well-controlled study should be substantiated by persuasive data from other well-controlled studies or studies showing the related pharmacologic effect. And this is why the mechanistic relevance of PET is so important. Now, granted, there will always be proprietary issues or concerns for the industry in their interaction with government or academic institutions, and as for any collaboration or liaison, these things must be negotiated and agreed. However, I think the one issue that I want to emphasize just now is the issue of, on the one hand, all of us are seeking cutting-edge projects, and the era of genomics will allow that virtually for everyone. And that has to be balanced against the level of validation necessary, first of all, for our internal business purposes, but then ultimately, and perhaps more importantly, also for any regulatory applications to which these data might be put. Bob Temple published back in 1995 an article about endpoints, and I'll just emphasize that he said there's nothing new about the use of surrogate endpoints. In fact, there are quite a few examples. On the other hand, also in his article, and I've expanded on some of the terminology that he used, we can go sadly wrong if we don't have full validation. And so there are a number of ways that we might make a mistake using surrogate endpoints, or even biomarkers in business decision-making. We need to be careful with the validation. However, I would emphasize also, draw your attention to the bottom line in your handouts, that it may well be in some cases that the PET allows access to a parameter which is not otherwise accessible, and therefore it might be that it's clearly superior to the existing clinical instruments. I will use Parkinson's disease as an example. If we are interested in a disease modification indication for Parkinson's disease, it's clear that the classical clinical endpoints are not suitable for this purpose because they're not able to distinguish between a symptomatic therapy and disease modification. In fact, there were some clever protocol designs and some rationalization which have gone into prior studies. I won't mention them in particular, but in fact mistakes can be made if we try to rely on, for example, the duration of the improvement in symptoms relative to the plasma profile of the drug. And so I would emphasize once again the validation is quite important, but finally the PET marker may well be superior to classical clinical endpoints because they get at the mechanism of action of the drug. Now I have just a couple minutes here, so I won't dwell on what I think are some of the criteria for a rational and efficient protocol design for phase 2, but I will welcome questions in that regard. I would emphasize, for example, that the use of fluorodeoxyglucose PET scans to stage cancer is something which is reimbursed by HCFA, and therefore it's clear that a certain level of validation must be there for HCFA to do this. And despite the presence, though, of a couple other very significant publications of data relative to this validation question, there is currently just now beginning a trial which is intended to validate FDG for the purpose of following the course of therapy. And so it's clear that the validation necessary for PET or any other technology to be used to follow the course of therapy is quite different than the validation necessary simply for staging cancer, for example. A few more criteria which are, I think, secondary to the criteria in the prior slide. And finally then, again, I will emphasize that using PET in the drug development program provides a continuum of value added from the non-clinical studies through phase 1, phase 2, 3, and right out into phase 3 and 4. And in fact, I would propose, this is my own form of words, which I've just splashed on the slide. I'm not particularly proud of these, and therefore I'm not wedded to them. I would be especially interested in any discussion. I think that the level of validation will obviously be greater as we progress through the clinical trials process. And so one question then is, how much validation is necessary in order to use, for example, something to stage cancer at the early stages of drug development? How much more validation is necessary to establish a tracer for mechanistic purposes? How much more validation, again, then to use as a confirmatory endpoint under the FDAMA criteria for a single phase 3 study? And then finally, what more validation will be necessary to establish this as a surrogate endpoint? I think this is a current topic of discussion, and I would welcome anyone's views in that regard. So, I'm grateful for the time that you've spent with me this morning. I hope we give you good value for the time you're spending with us. And I would like now to introduce Bill Moses. Bill took a degree in physics at UC Berkeley and is currently a physicist at Berkeley National Labs. He'll be telling us about the fundamental physics and hardware of positron emission tomography, which lends so much to the power of this technique. Bill?
As Richard said, I'm going to be talking about the imaging instrumentation for positron tomography. The tack that I'm going to be taking is that you're actually not all that interested in how PET imaging really works, but you're sort of interested in using PET as a tool in order to do some other stuff. And like all good scientists, in order to use a tool, you really have to understand how that tool works, what kind of data it's really giving to you, and what are the advantages and disadvantages of that particular tool. I'm going to start by sort of doing PET in 3 easy steps here, the basics of PET. Step 1 is that you inject the patient with a radioactive labeled drug. The drug is labeled with a positron emitter. Positron is an antiparticle of an electron. The drug localizes in the patient depending on what the metabolic properties of that particular drug are. This cartoon here shows that there are established ones for the brain, for the heart, for bones, for a number of different organs and a number of different diseases. One of the advantages of PET is that the amount of drug that you have to inject in the patient tends to be very small. Nanomolar type quantities are usually sufficient, and therefore it's truly a tracer technique. It does not actually perturb the system that you're trying to look at. It also means, since you're injecting a relatively small amount, it also means that the radiation dose tends to be reasonable. It tends to be sort of on the order of a diagnostic X-ray. I'm not really going to talk about the chemistry at all, but the isotope itself is physics. I'm a physicist, so I can talk a little bit about that. You certainly, of course, want if you're going to sit down and design what the ideal tracer isotope would have in it, you would want it to have interesting chemistry. You'd want it to be easily incorporated in the drugs that you're trying to use. You'd like it to have a half-life that's about an hour. The reason for that is that your maximum study duration tends to be about 2 hours for patient comfort. The patient can't really lie still for longer than about 2 hours, and you'd like the isotope to pretty much completely decay away during that 2 hours. However, you don't want too short of a half-life because you have to do some chemistry, and the chemists get really cranky when they're dealing with isotopes that are very short. You'd also, since it is a relatively short half-life, you'd like it to be, it needs to be produced relatively close by. However, with a 2 hour or with a 1 hour half-life, I'm from Berkeley, up San Francisco area. We occasionally get isotope from the Los Angeles area. Something that's an hour or so away by plane is considered local enough. And with those caveats, the isotopes that are most commonly used are these ones labeled over here. Of these, fluorine-18 tends to be the most common one. It's often substituted for hydrogen. We also have isotopic substitutions for oxygen, carbon, and nitrogen. And basically, with this in your armamentarium, there are very, very few biologically active compounds that you can't label with a positron emitter. Okay, step 2 is after the drug has concentrated, the radioisotope decays, emitting a positron. Now, that positron is going to find an electron in the tissue somewhere, and when the positron and the electron get together, they're going to annihilate and form a pair of back-to-back 511 keV gamma rays. Now, what you hope is that these two gamma rays will go through the patient and enter into a ring of gamma ray detectors that encircle the patient there and be detected. When you see two interactions at the same time in your ring, then you know that your positron-emitting isotope lies somewhere along the line that connects those two detected points. Now, what I showed you on the previous slide was basically a single ring encircling the patient. That will collect data from a single plane. In reality, you'd like to have a little bit larger field of view, and so what we often do is we stack up multiple planes on top of each other. Each of these, this would be one plane across here, and you will stack up multiple planes on top of each other. There are often tungsten septa that kind of they basically keep you looking at the radiation only in that one particular plane. They tend to eliminate radiation that is coming cross-plane or from at least large angles cross-plane. You can remove those septa. When you remove those septa, that is known as doing 3D PET, and I'm going to talk about that later in more detail. But basically what happens in a normal PET camera is that you take a number of 2D images, stack them up, and then form a 3D image. Okay, now the third step is to take the data that we have acquired and to reconstruct it using computed tomography. Now, what computed tomography does is it allows you to basically slice up an object mathematically. As an example, on the left-hand side here, I show an X-ray image of, it's not a medical object, it's actually an automotive transmission. But when you do that, all of the parts are sort of lying on top of each other, and although you can get very good resolution since everything is jumbled on top of each other, you can't really see very much detail in the center there. However, if you're able to take a single slice of that and then x-ray it, you would get the picture on the right, which is the sort of thing that you get with computed tomography, and that is an actual computer tomography image of a transmission, and you can obviously see the detail much easier. So basically with the computed tomography, it allows you to to separate individual slices and see the full amount of detail on each slice. Now the mathematics behind computed tomography is much too complicated for me to go into in this. It would take the whole hour to do that, but the basics behind it is described here. That if you have a two-dimensional object right here, if you take a one-dimensional projection of it, basically if you sort of do line integrals across like that, or you look at a projection across in that direction, what you will see is this figure down, something that looks like that down here. This portion of the shape is due to the circle right there. This sort of hole here is due to the lower activity there, and this bump over here is due to the higher activity there. If I do that same thing looking in a different projection, I will see those same sort of features. I see the overall big bump, the hole, and then the narrow bump, corresponding to those three different points in the image. I will state without proof that if you take projections in all sorts of different angles, that you have enough images or enough information to be able to figure out the shape of those various objects and their locations. And this slide here sort of shows the images that you can get with PET. This is an image of an epileptic patient taken both with PET and NMR, or MRI imaging. And it shows you the basic differences. With the NMR, you get a very high spatial resolution. You get sub-millimeter spatial resolution, but what it's doing is it's showing you the structure of that particular patient's brain, and you can't see anything unusual or anything wrong with that patient's brain from that MRI image. With the PET image, however, and this is actually a fairly poor quality PET image, you can see that this area over there is definitely got a lot higher activity than the rest of the brain. And so PET is showing you the function rather than showing you the structure. And the function that the resolution that it shows it with is approximately 5 millimeters. Okay, that is the basic fundamentals of PET. I'm going to talk now a little bit about the design of a typical PET camera. PET cameras are commercially available. Shown here are pictures of ones offered by GE Medical Systems and from Siemens CTI. Typically, they'll have about a 30-centimeter diameter patient port there. That's large enough to get a head in, not a whole body, or if they're whole body machines, which are much more common, they'll tend to have about a 50-centimeter diameter port in there. They'll have sort of a few dozen layers that cover about 15 centimeters axially, about 15 centimeters kind of in that direction. The spatial resolution is sort of 4 to 5 millimeters. It covers about 2% of the solid angle, so you get about 2% of the emissions coming out, and they cost a few $1 million. If you open up the inside of one of them and look inside, the things that is actually doing the gamma ray detection, in the early days you would have seen something like this. You'll see a scintillator crystal. A scintillator is a material that when gamma radiation interacts with it, it converts the gamma radiation into visible light. That scintillator crystal is connected to a photomultiplier tube. The photomultiplier tube then sees that visible light and converts it into an electrical signal. the detection of the gamma ray is really determined by the scintillator crystal. And the dimensions of the scintillator crystal determine its spatial resolution. So the distance in this direction, the ring is kind of going around like so, and the radioisotope is kind of over in that direction. The distance that way basically gives you your spatial resolution in plane. The distance of the scintillator crystal that way gives you basically your slice thickness, or the spatial resolution plane to plane. And then you tend to need a certain depth of scintillator crystal. The absorption of the gamma radiation is exponential, and you need about 3 attenuation lengths in order to get good efficiency for detecting that gamma ray. That's what early ones were like. Nowadays, what you will see is something that looks very much like it, has very similar performance, but is much more cost effective. You basically take a much larger block of scintillator crystal and partially cut it up into smaller crystals. You're very, very careful about how you cut these crystals. What you're trying to do is very carefully tailor the pattern of light that it makes on that group of 4 photomultiplier tubes. For example, an interaction in this corner crystal here will put a lot of light into this photomultiplier tube, but not very much light into the other 3 photomultiplier tubes over here. However, an interaction in this crystal right here will put sort of equal amounts of light into those photomultiplier tubes and not very much light into these photomultiplier tubes. And basically, so by taking the ratio of the amount of light